To form gametes or sex cells, a process called meiosis divides a somatic cell into gametes.
Wait, hold on.
Both of Mendel's laws serve as the basis for meiosis. The law of independent assortment states that each pair of alleles segregate independently during gamete formation. And the law of segregation states that two alleles segregate during gamete formation into different gametes. After interphase occurs, meiosis 1 begins with prophase with a tetrad of DNA. Prophase 1 is the longest stage of meiosis, and chromosomes begin to condense in the nucleus as the nucleoli dissolve. Homologous chromosomes, which are chromosomes of the same length, centromere position, and gene loci, begin to pair up. Crossing over occurs, which is when genetic material is exchanged between homologous chromosomes. Chiasmata, regions where crossing over has occurred, are present in each homologous pair, and the microtubules at the mitotic spindle become visible. Later in prophase 1, the kinetochores attach to the microtubules in each homolog, and the homologous pairs begin moving to the metaphase plate. During metaphase 1, chromosomes are now fully condensed and visible, and the pairs of homologous chromosomes line up at the metaphase plate. Both chromatids on one homolog attach to kinetochore microtubules from one pole, and the other homolog chromatids attach to kinetochore microtubules from the other pole. At anaphase 1, cohesins cleave the homologous chromosomes apart, and they are guided by the spindle apparatus to opposite poles of the cell. Sister chromatids are still attached by a centromere, and they move together to the end of the cell. Telophase 1 and cytokinesis occur at the same time and form a cleavage furrow in animals or cell plate in plants, making two haploid cells. No replication occurs after this stage. Meiosis 2 begins with the two haploid cells from the end of meiosis 1. In prophase 2, the spindle apparatus forms, and the chromatids move towards the metaphase 2 plate. In metaphase 2, the sister chromatids bound by centromere line up at the metaphase plate and kinetochores attached to the centromeres in the microtubules. Because of crossing over, these chromatids are no longer genetically identical. In anaphase 2, the centromere is cleaved to separate the sister chromatids, which move to two opposite poles. Finally, in telophase 2 and cytokinesis, the chromatin decondenses, making four haploid daughter cells that are all genetically different from one another. Other four cells are one other.